Welcome to the second lecture, Renal Physiology. Today we will talk about kidneys' role in maintaining homeostasis. And before we will begin, let me remind you once again that the lecture you find in Facebook group under Files category. And at the very beginning, I would like to tell you the quote which I really like, that the composition of plasma is determined not by what the body ingests, but what the kidney decides to retain and what they excrete. This sentence really brilliantly describes the role of kidney in the body. And the learning objectives today's lecture will be: we will talk about humoral and humoral urine formation, then we will discuss the micturition, its mechanisms and control. And finally, we will talk about the role of kidney in regulation of arterial blood pressure, blood osmotic pressure, ECF volume, ion composition, and blood pH. So the micturition reflex, that is what is known as discharge of urine from urinary bladder. In your books, you can find it as micturition or urination or urine voiding. Renal excretion of water is referred to as diuresis. And the micturition reflex, it occurs where the combination of voluntary and involuntary muscle contraction and a lack of voluntary control over the micturition. That is what is called urinary incontinence. Let's talk about micturition. The desire to urinate will be stimulated by distension of the urinary bladder walls, and the first sensation will occur when the urinary bladder will be filled up to about 150 ml, and when it reaches about 300 ml of urine or more, then the sensation of uncomfortableness occurs. And the maximal capacity of the bladder, it will be around 500 ml. But in the end of the urination, still less than 10 ml of urine is usually remains in the urinary bladder. So here you can see the sequence of events which will occur in urinary bladder and how micturition occurs. So when volume of fluid in bladder will reach or will exceed 150 ml, it will lead to expansion of walls, which will activate stretch receptors, and impulses will be sent to the spinal cord. Here you can see the centers and the location. And what happens in spinal cord? These impulses, they will decrease the sympathetic activity, which will cause relaxation of internal urethral sphincter and will lead to its opening. And it will also increase parasympathetic activity, which will lead to contraction of the detrusor muscle. But also, at the same time, impulses will be sent to the brain cortex, and it will lead to decreased somatic control and relaxation of external urethral sphincter. And as a result of opening of external internal sphincter and contraction of micturition occurs. That's the complete pathway, including the brain cortex on top, which will control the micturition. Let's talk about mechanisms of regulation of those three steps of urine formation which we mentioned last lecture. So mechanisms of regulation, as always, you have neural and humoral. It will be done through the ANS. And as you might remember, for the kidney, mostly we will discuss the sympathetic control, which will mostly exert its influence on the renal blood flow, means it controls the glomerular filtration because vasoactive substances will be released. Oral control will affect not only filtration, glomerular filtration rate and renal plasma flow, but also will affect reabsorption and secretion. And here you have the hormones which you previously studied, and today we will revise their functions. So let me remind you main principles of glomerular filtration across the filtration barrier. You have one major force which favors filtration, which is the capillary hydrostatic pressure. And then you have two forces which oppose the filtration. One is the colloidosmotic pressure of the capillary or oncotic pressure of the capillary, and another one is hydrostatic pressure of the primary urine from the Bowman's capsule. And the resulting force, which is the net filtration pressure, will determine whether you will have filtration, whether it occurs or not. Here you have the sample of pressures with the resulting one, which is equal to 16. So those things which we've mentioned last time and those which will affect the pressure inside the glomerulus and the glomerular filtration rate. So the first example here you have constriction of afferent arteriole, which could be done as a result of sympathetic activation, interaction with alpha-1 adrenergic receptors. This will reduce the blood flow. As a result, hydrostatic capillary pressure decreases and glomerular filtration rate drops. In case of constriction of efferent arteriole, you will have rise of resistance, which increases the hydrostatic pressure in the capillary and increases the glomerular filtration rate. And this will happen to the low levels of angiotensin present in blood. And if you will have dilation of efferent arteriole, the opposite occurs. With dilation of afferent arteriole, the blood flow increases, as you remember. The hydrostatic pressure in the capillary will increase. This will increase the glomerular filtration rate. So how to maintain healthy GFR? Kidney, they have perfect mechanisms of autoregulation. This will ensure that the glomerular capillaries will remain safe in case of increasing blood pressure, and also this will provide the necessary pressure which is needed for the glomerular filtration to occur. And here you have two mechanisms which we'll discuss: the myogenic response and tubulo-glomerular feedback. And before we will begin, let me remind you the structure of juxtaglomerular apparatus, which consists of granular cells, which are also called juxtaglomerular cells. They surround the afferent arteriole like a cuff around the small muscles of the walls of the afferent arteriole, and they're responsible for the secretion of renin, as you remember. Then you also have the maculodenser cells in the distal tubule, and you also have extraglomerular mesangial cells. So the first case which we will talk about is the myogenic response. Actually, you had that when you were studying the smooth muscle tone in blood vessels. So myogenic response, it's a result of direct reaction of the vascular muscle to a stretch or relaxation, most commonly to the stretch. So what happens in this case? If mean arterial pressure increases, it leads to stretching of smooth muscles in the afferent arterial wall. And in this case, due to the stretching, calcium channels will be opened and calcium rushes in. And as you remember, you need calcium for the contraction, and that's what leads to vasoconstriction. And the opposite is also. So why do you need it? If you will have rise in pressure, it will be counteracted by this vasoconstriction, and it will offset the rise in pressure, which will protect the glomeruli from short-term fluctuations in blood pressure, and it will keep the changes of renal plasma flow and thus the glomerular filtration rate very small, which is very important. The second case is the tubulo-glomerular feedback. It happens inside the kidney as well. and that's the feedback from the tubules back to the glomerulus. So let me give you an example. If glomerular filtration rate decreases, then the flow of urine through the decreases, which means there will be less, the amount of sodium which escapes reabsorption will be less, and less sodium flowing past macula densa will lead to renin release, which will further cause angiotensin production. That's one mechanism. And as you remember, we've mentioned that angiotensin 2 leads to constriction of the efferent arteriole, which will increase the hydrostatic pressure in the glomerulus. On the other hand, you will have paracrine secretion from maculodensa cells of vasoactive substances, which will lead to the dilation of afferent arteriole, which increases the blood flow through the glomerulus and also increases the hydrostatic pressure in it. And in the end, you have increased in GFR. So GFR is back normal. And the opposite situation is also true. So tubulo-glomerular feedback. Maculodensa cells, they respond to changes in the concentration of sodium chloride passing by the distal convoluted tubule. And if it decreases, it will lead to vasodilation of the afferent arteriole. And you remember that vasodilation of the afferent arteriole will result in increased blood flow to the glomerular capillaries, increased glomerular filtration pressure, and of course they increase the glomerular filtration rate. And the opposite, if the sodium chloride concentration increases, is also true. So as you can see, this graph, it nicely shows that autoregulation of renal blood flow and glomerular filtration rate will be independent within the wide range of pressure fluctuations, actually from 60 to about 180 millimeters of mercury. You will still manage to maintain a healthy glomerular filtration rate around 180 liters per day. Humoral control will be done by the following hormones. The functions, actually you've studied during the first SEM, and here you have the site of action, but we will still revise the action of main the hormone. So, urine formation, it goes through the following mechanisms. You have glomerular filtration, obligatory reabsorption in PCT, then you will have osmotic concentration of urine in tubular system, and then facultative reabsorption and secretion in DCT, and then final concentration of urine in collecting ducts, urine excretion, and micturition. And these three steps will provide the following functions of the kidney: regulation of osmotic pressure, regulation of blood pH, and CO2 through the pH regulation, then nutritive function and blood pressure regulation. So control of salts and water excretion. Why do you need it? It helps to maintain a body fluid volume, maintain an osmolarity of that fluid, and maintain the arterial blood pressure. How to easily remember? There is a 60-40-20 rule. What does that mean? Total body water in adults in liters is about 60% of the body weight in kg. Intracellular fluid, the ICF, is about 40% of the body weight, and extracellular fluid, the ECF, is about another 20% of the body weight. You can remember it easily: 60-40-20 rule. So, body fluid compartments. It will be slightly different in female and male. And what do you have? You have total body fluid, as I've mentioned, which is subdivided into intracellular and extracellular. Intracellular about 2/3, extracellular about 1/3. And extracellular fluid will be further subdivided into interstitial and plasma. Beside that, you have small amounts of transcellular fluid, which could be found like cerebrospinal fluid, synovial fluid, aqueous humor, pleural, pericardial, and other cavities fluids. So how do we gain and how do we lose water? Here you have a summary on water gain and water loss. So water gain, here you have small amount of metabolic water, then with ingested foods and ingested liquids. How do you lose water? Mostly through kidneys, as you can see, about 1.5 liter per day. Then skin, but that depends on environmental conditions, the lungs while breathing, and small amounts through the GI tract. And you should remember that normally body fluid volumes, they remain constant because, as you can see, water gain is equal to the water loss. Dehydration occurs when water loss is greater than water gain. When this can happen? During strenuous exercises, diarrhea, for example, in case of GI tract infection, in case of vomiting, and you can also have hyperhydration. It occurs when water gain is greater than the water loss. When this can happen? Consuming too much of water, intravenous fluids are given but not balanced in composition to match fluids and electrolyte loss. So these are the main definitions you need to know before we will proceed. So better understand fluid shifts between compartments, you have to refresh few basic principles. ICF and ECF are in osmotic equilibrium, means the osmotic pressure is exactly same. Sodium is the major cation in the ECF, and potassium is the major one in intracellular fluid. The distribution of sodium and potassium will be maintained by sodium-potassium pump, and equilibrium between the ICF and ECF occurs through the water movement, but not movements of elective particles. And fluids they will move unassisted across cell membranes only because of the osmotically pressure differences. So let's talk about dehydration first. You can see dehydration will lead to decreased flow of saliva, which will create the sensation of dry mouth. Then it will lead to increased blood osmolarity, which will stimulate osmoreceptors in hypothalamus. Then it will also lead to decreased blood volume, which will decrease the blood pressure, and it will increase the activity of renin, angiotensin, aldosterone system. And all of these will stimulate thirst center in hypothalamus, will increase your thirst, and as a result your behavior will change, water intake increases, so to compensate for the water loss. Once again, normally body fluid volumes remains constant because water loss equals water gain. Mind you, these are the cases, some of the cases when dehydration might occur. And the precise amount of pressure required to prevent the osmosis is called the osmotic pressure. That's what you've learned before. And osmolar concentration of solution is called osmolality when it's expressed in kg, in osmoles per kg. And it will be called as osmolarity when it's expressed as osmoles per liter of solution. And here you have a summary of osmotic pressure regulation through the hypothalamus. So here you have two cases. If there will be, for example, low water intake, it might lead to dehydration. As a result, plasma osmolarity increases. This will stimulate the osmoreceptors in hypothalamus, which will produce more of ADH, which will reach posterior pituitary and will be released into the bloodstream. More ADH will reach kidney, which will cause increased water reabsorption. And as such, less water will be excreted in urine. That's the negative feedback correction. And the opposite is also true. When there is an overhydration, plasma osmolarity is less ADH is secreted, so no water will be reabsorbed, because you remember, without ADH, aquaporins will be formed and water is not reabsorbed and it will be excreted in urine. And here are the few definitions which you studies. Anuria, that's when abnormally low urine output, less than 50 ml per day. Polyuria, that's when output is below 3 to 500 ml per day. And polyuria, I guess you do remember this symptom from diabetes, that's excessive or abnormally large production of. And why it's important to control volume and osmolarity? Because it might affect the blood pressure as well. And which systems major to have to control? You have baroreceptor control and you have osmoreceptor control. Let me guide you through these two systems. In case of baroreceptor control, what you will sense? You will sense the effective circulating volume. And sensors means receptors will be located in carotid arch and also in other arteries and also in atria. What are the afferent pathways? Will have both neural and humoral control, and they will be divided into long term. The short term, of course, it's the neural one from carotid sinus and aortic arch, for example, through the medullary center back to the heart and vessels. And the blood pressure will change as a result. The long-term one, that's the humoral one, and it will act through kidney. One of the examples, it will affect the sodium excretion. Osmoreceptor control, that's what we have just discussed. It depends on plasma osmolarity. And you will have hypothalamic osmoreceptors. And here you have at least ADH and thirst. ADH, as you remember, acts through kidney and it affects the water excretion. And thirst, it will change your drinking behavior and it will increase water intake. Now, let's talk about humoral control of renal and chloride reabsorption, because that's what also affects the osmolarity. In situation when there is increased intake of sodium chloride, that's when you eat some salty food, for example, that might lead to increased plasma osmolarity because sodium chloride concentration increases, and it will lead to water retention, increased blood volume. What happens in this case? Increased blood volume will lead to stretching of the right atria, and that's where the sodium natriuretic peptide will be produced, which acts on kidney, and it reduces the reabsorption of sodium by kidney. As a result, sodium will be lost, and since it's a specifically active substance, water will be lost as well, and it decreases the blood volume. On the other hand, you will have deactivation of the aldosterone system, which will also reduce the reabsorption of, because decreased production of aldosterone happened. So, elimination of excess body water or solutes occurs mainly by control of their loss in urine. And the extent of urinary salt loss is the main factor which body fluid volume, because it is an osmotically active substance. And the reason for that is that water follows solutes, osmosis. And sodium and chloride, these are the two main solutes in extracellular fluid. So once again, the main factor which determines body fluid osmolarity is the extent of urinary water loss. Now, here is the summary of factors which will maintain the body water balance. First, center in hypothalamus, drinking behavior. Then angiotensin, we will talk about that, stimulates secretion of aldosterone and reduces water loss. Aldosterone by itself, by promoting reabsorption and thus increases well. Natriuretic night, which promotes natriuresis, means full of sodium, and increases loss of water in, and antidiuretic hormone, which will promote insertion of water channel proteins known as aquaporins into apical membranes of principal cells and collecting ducts in the kidney, and thus it increases its permeability for water, causing water reabsorption, and as such it reduces loss of water. Now, one more case which I would like to discuss, water intoxication. What's that? If there will be excessive water loss, like sweating, vomiting, or diarrhea, coupled with intake of plain water, like that's what is known as tap water, it will lead to decreased sodium concentration of interstitial fluid and plasma, which is known as hyponatremia, and it will lead to decreased osmolarity of interstitial fluid and plasma, and as a result, osmosis of water from interstitial fluid into the intracellular fluid occur. That is what is known as water intoxications. Cells, they will swell, and some of them might get damaged. And the most sensitive cells will be like cells of the nervous system, might lead to convulsions, coma, and possibly death. And prevent these in cases of severe electrolyte and water loss, solutions are given for intravenous or rehydration therapy. They should be balanced on electrolyte content to avoid water intoxication. So let's talk about renin-angiotensin system because we have mentioned it already a couple of times. So here you have a summary on factors which will activate or inhibit this system. Activation of this system could be done by increased sympathetic activity, increased circulating catecholamines and prostaglandins, and high concentration of angiotensin and vasopressin inhibits this system, as well as increased arterial pressure and increased right pathway across the macula densa. So here is one of the possible summaries on renin-angiotensin aldosterone system. You have dehydration or sodium deficiency or any other reason which reduces perfusion of kidney. That could be decrease in blood volume or blood pressure. As a result, there will be activation of juxtaglomerular cells of kidney, and they will start producing renin. Renin converts angiotensin 1, which will go to the lungs. In the lungs, you have angiotensin enzyme, which will convert angiotensin 1 into. And the second one will have multiple effects, such as vasoconstriction, which increases the blood pressure until filtration backs to normal, or it acts on the cortex of the suprarenal gland, produce aldosterone, which causes absorption of sodium and water retention, which will increase the blood volume, and this increases the blood pressure as well. And here you have even more effects of angiotensin. Help to maintain a healthy blood pressure, means healthy renal plasma flow and healthy GFR. So increased sympathetic activity, which will lead to vasoconstriction, reabsorption of and water retention. Angiotensin can do it by itself or through aldosterone secretion. Then angiotensin is a very powerful vasoconstricting substance which will lead to vasoconstriction, and also it stimulates ADH secretion, which in turn leads to reabsorption. And all of these will provide effective circulating volume, then perfusion of the glomerular apparatus increases, returns back to maintain glomerular filtration rate. And this is a nice summary on overall kidney control. Here you have both intrinsic level and extrinsic level. Intrinsic level, that's what we have discussed, the myogenic control, like stretching, increased calcium influx, contraction, which is known as vasoconstriction. Then, also intrinsic one, the tubular-glomerular feedback. If less sodium chloride passes through maculodenser cells, they will activate both angiotensin and paracrine secretion, which will lead to vasodilation, and healthy GFR will be restored. And extrinsic mechanisms, they consist of two, the humoral and neural control. Humoral, it's almost entirely about renin, angiotensin, aldosterone mechanism, which we have just discussed. And the neural, it's mostly about the sympathetic control of the vessels. Besides renin-angiotensin system, you also have kallikrein-kinin system. That's when finally you will have the bradykinin produced. That's the degradation product. And the final effect depends on whether it binds to 1 or 2 second receptor. If it binds bradykinin receptor 1, it leads to vasoconstriction. If to the second one, then vasodilation. Kallikrein-kinin system, it's related to polypeptides such as bradykinin, as I've mentioned. And it has multiple roles in the body. I guess you will be given more details in biochem. And it's responsible for the blood pressure control and functions. In most of the, it causes vasodilation. It will increase secretion of prostaglandins, and it decreases the vasoconstriction by angiotensin. And one more system which also helps to restore the blood flow through the kidney is known as prostaglandin system. Derived from fatty acids, these are hormone-like substances. Most of them lead to vasodilation, which will increase the perfusion in kidney. So here you have the scheme how to get prostaglandins. Like you can have PGI2. Effects could be different. And here you have the summary, the effects. So let's move electrolytes and their functions in the body and control of electrolyte levels in the body. So some general functions. They help to control the osmosis and thus the balance of water between CF and ICF. It helps to maintain the acid-base balance. It carries the electrical current, which allows the production of action potentials, and it serves a cofactor needed for optimal activity of enzymes. So here you have a summary on main differences in electrolytes concentration in extracellular fluid like blood, plasma, and interstitial fluid, and in intracellular fluid. So as you can see in red, that's the blood plasma, and like here you have, and interstitial fluid in blue. Yellow is the intracellular one. And as I've mentioned, sodium found in, and it's very low in intracellular fluid, while potassium is very high inside the cell. And so it goes for the rest of the electrolytes, as you can see. And this scheme, I guess, it helps remember the ions a bit easier. Here you have the ECF and here you have the ICF. And as you can see, major electrolytes in extracellular fluid, you have the sodium and chloride and bicarbonates, while inside the cell you have potassium and magnesium, and you have phosphates and organic anions. So that's the summary on distribution of electrolytes in and out of the cell. So let's talk about some of them. It is about 90% of the ECF, and normal plasma concentration is as follows. It provides the osmolarity, as we've mentioned, of the ECF. And as you might remember from excitable tissues, it's responsible for the generation and conduction of the action potential in excitable tissues. As we've mentioned already, kidney, they will excrete excess of sodium, but they also can retain it. And levels of sodium in the body will be controlled by aldosterone and ethyl natriuretic peptide. And two conditions might occur. They are known as hyponatremia low and hypernatremia high. Chloride ions. It is also most prevalent anion in extracellular fluid. Here you have the normal plasma concentration. Chloride moves relatively easy between the compartments, and it helps to balance the levels of anions in different fluid compartments. I hope you do remember the chloride shift in RBCs and plasma. It's related to the transport of carbon dioxide. Also, you need chloride ions for the production of hydrochloric acid, and it's secreted in the gastric juice. You do not really have a hormone which is responsible for the regulation of chloride, but it is really connected to the sodium regulation, and two might develop hypochloremia and hyperchloremia. Potassium ions. That's the major cation in intracellular fluid, and that's the normal plasma concentration. You can see very low. Potassium we discussed when we were talking about resting membrane potential, and also it is involved in polarization phase of action potential in excitable tissue. It also helps to maintain the normal intracellular fluid volume and also helps to regulate the pH of the body fluid. It moves in or out of the cells, and it will be often exchanged for the hydrogen, called the pH. And the hormone which controls the aldosterone, it's responsible for the secretion of potassium. And two conditions might develop: hypokalemia and hyperkalemia. Why do you have to remember about potassium, and why is it important to keep it low outside the cell? Because serious manifestations which occur when the serum potassium is more than 7, that's what is known as hyperkalemia. It might lead to cardiac manifestations, and how you can recognize it: by tall peak T waves with a shortened interval. That these are usually like the first findings in the ECG. Here you can see usually wave is not that peaked and not that tall. So here you have a summary on potassium regulation. If potassium intake, for example, increases, leading to increased plasma concentration, then aldosterone stimulates secretion of potassium in the collecting tubules and leads to potassium excretion. And here you have like potassium is filtered, reabsorbed, and secreted by the nephron to gain the normal concentration of potassium within the blood plasma. And here you have a summary on potassium handling. Here you have the factors which lead which increase potassium entering the cells, like alkalosis, and factors lead to potassium efflux. It leaves the cell, like insulin, acidosis, destruction of cell because of iron, and sodium potassium pump inhibitors, because sodium potassium pump, it pumps potassium into the cell. The next one is the bicarbonate ions. That's the second most prevalent extracellular anion. Here you have the normal plasma concentration, and remember we mentioned the chloride. It exchanges for it, which will help to maintain the correct balance of anions in CF and ICF, and it's responsible for blood pH regulation. Calcium ions. That's the major cation in the intracellular fluid. And normal plasma concentration, here you have two fractions. You have free unattached calcium and the one which is attached to various plasma proteins. And as you can see, about 98% calcium in adults is located within the skeleton and teeth combined with. In body fluids, calcium is mainly in extracellular fluid and of course for the hardness of, but it also plays an important role in blood clotting. That's the factor number 4. Then also essential for the neurotransmitter release in synaptic transmission. It will not be released unless calcium will enter the presynaptic part. Then it's responsible for the maintenance of muscle tone, muscle contraction, and excitability of nervous. And calcium ion concentration will be controlled by parathyroid hormone. PTH increases on resorption, then so reabsorption of calcium in kidney and absorption of calcium in the GI tract. And for that also better need the vitamin D3. Calcitonin produced by the thyroid gland, it inhibits the activity of osteoclasts, accelerates the calcium deposition into and thus lowers the blood levels. And here you have conditions hypocalcemia and hypercalcemia. And another ion which is coupled to the calcium, that's the phosphate. You have the normal concentrations, and it's important buffering capacity in blood and in urine. And most phosphate ions, they are bound to organic molecules such as lipids. You have phospholipids, then you have protein, and also the substance which is as ATP. And for phosphate ions, you have parathyroid hormone PTH, which stimulates reabsorption of resorption of bone matrix by osteoclasts, which releases both phosphate and calcium into the bloodstream, but it inhibits reabsorption of phosphate ion while stimulating reabsorption of calcium ion by the renal tubular cells. And calcitriol promotes absorption of both phosphates and calcium from the GI tract, and to develop if it's below or above normal blood. One more ion, it deserves to be mentioned, magnesium ions. It's about 50% of the body total body magnesium. That's the part of the bone matrix, and remaining one, it occurs as magnesium ions in cellular and mostly and very little in extracellular fluid. Is the second most intracellular ion. And why do you need to remember about magnesium? Because that's the cofactor of certain enzymes needed for the metabolic activity of carbohydrates and for the sodium potassium pump. And also it is essential for normal neuromuscular activity, synaptic transmission, and mostly the myocardial functioning, like secretion of parathyroid, regulation of blood plasma level of magnesium. It varies, and it's connected to the calcium reabsorption in thick ascending limb. And you can see that kidneys increase urinary excretion of magnesium in response to hypercalcemia, or hypercalcemia inhibits magnesium reabsorption, and the opposite is, and two conditions might develop. if it's below or above the next question, which we will discuss, is the regulation of blood pH. Remember that pH, that's the rigid constant, and fluctuations are known as acidosis when it drops below, or alkalosis when it increases greater than. But still, the fluctuation range is still very narrow, and as you can see, if it exceeds those small limits, it might lead to death. Few definitions to refresh the pH regulation. An acid, it acts as a proton donor, which is a molecule or ion that can contribute hydrogen ion to a solution. And a base, it's a proton acceptor or ion that will combine with the hydrogen ion to remove the solution. Hydrogen, that's the acid, and A minus, it stands for the conjugate base in hydrogen anion and conjugate acid-base pair. The strength of an acid is defined with respect of the ease with which hydrogen is released, and the strength of base is defined with respect of how strongly it binds hydrogen. So hydrogen, often we are exposed to increased hydrogen concentration. We have three mechanisms for the hydrogen removal. It will be the buffer systems, then excretion of carbon dioxide, and kidney excretion of hydrogen. Buffer systems, that's what you have in blood. Buffers, they act quickly to bind hydrogen, removing the highly reactive excess of hydrogen in the solution. Exhalation of carbon dioxide. By increasing the rate and depth of carbon dioxide can be exhaled. Within minutes, this will reduce the level of carbonic acid in blood, which rises the blood pH, reducing the blood hydrogen level. And kidney excretion of hydrogen. That's the slowest mechanism, but the only way to eliminate acids other than carbonic acid, as through the excretion means from the body. So let's refresh the buffer systems. It consists of a weak acid and the salt of that acid, which functions as a weak. Principal buffer systems in the body fluids are, you have four major buffers, which are protein and hemoglobin. Hemoglobin, that's the strongest one. And also you have a bicarbonate buffer system and phosphate buffer system. Carbonate buffer system, that's for the extracellular fluid, and phosphate, that's for the intracellular fluid. And here you have the equation, and where it shifts, because it works both sides, it depends on whether you place it into the acidic or alkaline medium. It will either accept excess of hydrogen, it will donate hydrogen solution. Now let's talk about regulation of blood pH by the respiratory system. For example, if blood pH decrease when it becomes acidic, it means when you will have increased hydrogen content. This will stimulate chemoreceptors. Remember that we have central and peripheral chemoreceptors. Central will be in medulla, and peripheral they are located in sinus and aortic arch. And impulses, they will reach the inspiratory center, which is in the medulla. Inspiratory area in medulla will be activated, and nerve impulses will be sent to the effector. And as a result, the rate and depth of will increase, and more CO2 will be exhaled, which means as less carbonic acid will be produced, there will be fewer hydrogen ions present, and blood pH will increase, hydrogen, and then the negative feedback will be given. So that's how lungs and the respiratory system help to control the blood pH. Once again, an increase in the carbon dioxide concentration fluids increases hydrogen cerebrospinal fluid and thus lowers the pH. And that's what leads the respiratory system and finally leads to exhalation of more of CO2 and lowers the hydrogen blood. And then the negative feedback. What are the possible acid-base disturbances which might happen? If it's the respiratory system responsible, then two situations might happen: respiratory acidosis and respiratory alkalosis. Respiratory acidosis is caused by hypoventilation, resulting in pH and slight increase in bicarbonate. And here you have the values of partial pressure of CO2. Respiratory alkalosis is, on other hand, is caused by hyperventilation, which decreases the CO2 levels, resulting in increased pH and a slight decrease in bicarbonates. And possible causes include hyperventilation or traveling high altitude. Remember, during the classes we discussed amount. And since you remember that kidney is also responsible for the acid-base balance control, kidney also help to control the excretion of wastes, and if there will be any disturbance related to it, will lead to metabolic acidosis or metabolic alkalosis. Metabolic acidosis is caused by a gain in a fixed acid, loss of bicarbonate, and decreased pH. And possible causes include diabetic ketoacidosis, like methanol poisoning. Metabolic alkalosis is caused by loss of hydrogen as a fixed acid, which result in increase in bicarbonate and increased pH. Metabolic alkalosis is diagnosed when bicarbonate concentration is greater than 24. And possible causes include vomiting, when there is a hydrogen loss, or diuretic abuse. So how do kidneys control excretion of hydrogen? Here you have the scheme which illustrates how secretion of hydrogen. Hydrogen is secreted. Here you have the tubular lumen and the blood artery and intercalated cells in the collecting ducts. Hydrogen is secreted into the tubular fluid in both the PCT and collecting ducts. So what do you have? In proximal convoluted tube, you have hydrogen antipode. What happens in this case? Hydrogen, they will secrete the hydrogen as they reabsorbed. Hydrogen reabsorbed. In collecting ducts, apical membranes of some intercalated cells, they have a proton pump, hydrogen, like you can see here, which will secrete hydrogen to the tubular fluid in case when it's needed to be secreted. And and the mechanism you can see here, what happens in red blood cells when it transports carbon dioxide, or in parietal cells when the hydrochloric acid. You have carbon dioxide combines with water in presence of carbonic hydrase, and you will have carbonic acid, which is unstable, and it quickly splits into hydrogen bicarbonate, and hydrogen will be secreted into the lumen, maintain pH, be removed from the body, and bicarbonates, they will be absorbed back into the blood, maintain pH of the blood in exchange of chloride ion. Remember that chloride shifts so that the charge, the final charge of the cell will not change. Anion, and anion should go back in. And when hydrogen is into the fluid, of the collecting ducts, it has to be buffered. And what are the options? It could be buffered by phosphate ions, like you have the example of in here, buffering of hydrogen. And then it could be the ammonia and also the bicarbonates. So these ions, they cannot diffuse back into the tubular cells, and thus they will be excreted in urine. Thank you for your attention.